Bom dia a todos. Desculpa esse pequeno atraso. As apresentações serão em inglês, mas essa primeira introdução eu vou falar em português, porque a maior parte é brasileiro, e tem muito de Portugal também que fala português, então acho que todo mundo vai entender. Eu fiz só um resuminho do que nós vamos fazendo desde 2019. Em 2019, nós fizemos o primeiro workshop, que foi sobre Drug Discovery Development, com o professor Raymond Chinazzi. Neste evento, nós tivemos 211 participantes. Em 2020, nós fizemos o primeiro webinar sobre cultura de células 3D. 156 pessoas se interessaram pelo assunto. O Dr. Glauco Souza, que fez a apresentação, e isso nos motivou, em 2022, fazer um outro workshop que teve teórico e prático para poucas pessoas, porque a gente não é lógico por causa da logística, né? Mas a parte teórica a gente divulgou para todo mundo, inclusive a parte prática, nós tentamos dar pelo menos os POPs para as pessoas que participaram. E esse ano, para nossa surpresa, nós tivemos 388 inscritos no nosso workshop. Vai ser teórico, e o objetivo é mostrar a aplicação prática de alguns pesquisadores no mundo que fazem cultura de células 2D e 3D de uma forma mais avançada para cultura de células, transplante e outras aplicações. Desses, aqui só para vocês terem uma ideia, nós tivemos pessoas de várias instituições, tá? E inclusive de outros países, tá? E o nosso programa, e assim, eu queria agradecer primeiro também a todos vocês que estão participando, que nos incentivaram, então, a realizar esses workshops. Muito obrigada. E aos palestrantes também, que dedicaram tempo para preparar, né? A Natália Pelisson, que vai falar pela Greiner e vai falar a parte metodológica da cultura células 3D. A seguir, o professor João Ferreira, ele é de Portugal, vai falar, ele nos enviou um vídeo, porque, infelizmente, ele está em férias, mas foi muito gentil nos mandar um vídeo sobre a apresentação dele, que nós vamos passar. Depois, a professora Leda Bassiti, que está presente aqui conosco, da Emory University, e o Dr. Adam Grax, que é amigo da doutora Leda Bassiti, que também faz cultura de células, organoides, e também vai nos contar um pouquinho sobre a experiência dele. Amanhã nós teremos a doutora Marluce Mantovani, da Nucel, o doutor André Barateiro, que na verdade foi o principal motivo da gente fazer esse workshop. Ele é aluno de pós-doc do ICB, procurou o professor Flávio Galvão para fazer uma parceria. O professor Flávio, ele vai falar um pouquinho daqui a pouquinho, ele da cirurgia, e aí ele pediu ajuda para conseguir fígados para desenvolver a metodologia que ele vai apresentar amanhã. Nós teremos também a apresentação do Dr. Glauco Souza, que desenvolveu um método de cultura de células 3D, que será apresentado da Greiner. E, por último, Eleftherios Michalaides, que vai falar um pouquinho sobre trabalho, metodologias para estudar hepatite B, que é um dos interesses meu, da Michele, da Leda, e acho que muitos outros de vocês. Dr. Glauco, ele é brasileiro e desenvolveu a metodologia da Greiner, né? E ele resolveu ficar nos Estados Unidos para empreender. Depois, uma das minhas perguntas é: nos Estados Unidos, será que é mais fácil? Porque dá a impressão que todos que abrem startups, companhias de biotecnologia, acabam migrando para fora para expandir. Eu não sei se depois, a da Gabriela, que é do CITIC, que é o Centro de Inovação e Tecnologia do Instituto Central, ela vai estar conosco, né? E ela faz parte do grupo também para nos ajudar, caso se desenvolva um produto, para a gente poder colocar no mercado ou empreender também, né? Então, essa é uma pergunta que depois acho que vou fazer para o Dr. Glauco. Será que nos Estados Unidos é mais fácil, né? Ou o que será que tem aqui no Brasil que todo mundo acho que acaba migrando, né? E, por fim, aqui os coordenadores, tá? Eu e a Leda temos interesse na metodologia para estudar antivirais. O Flávio e a Márcia são do grupo de transplante. O Flávio vai falar um pouquinho mais sobre o interesse dele. E a Gabriela, do Centro de Inovação de Tecnologia do Instituto Central, que tem nos ajudado a entender um pouquinho melhor sobre empreendedorismo, startups e inovação. Por último, queria agradecer à FAPESP, ao CITIC e ao CNPq, que nos ajudaram parcialmente na realização deste evento. E, por último, eu queria pedir para vocês acessarem pelo QR Code, só para registrar a frequência, e depois a gente pode emitir certificados e também a gente precisa apresentar um relatório para a FAPESP. Aí eu agradeceria se vocês pudessem fazer esse favor. É só botar o nome, já captura o e-mail e registra a frequência fácil, fácil. Obrigada. Vou passar a palavra para o Dr. Flávio Galvão.
Bom dia a todos. É uma satisfação tê-los aqui no nosso workshop. Eu queria iniciar dizendo que tenho um grande orgulho de estar participando com a professora Suzane desse workshop. Ela que realmente é uma colaboradora de longa data. Nós já colaboramos em outras empreitadas aqui na Faculdade de Medicina. Temos um curso de pós-graduação que foi um dos primeiros a ser pós-graduação online. E agora a gente está muito orgulho de estar participando com ela nesse programa de workshops, né? Representando o INSS P7, que eu coordeno, e queremos dizer que somos muito agradecidos, professora, pelo seu trabalho, muito agradecidos. Eu queria dizer que é muito fácil colaborar com você. Aliás, às vezes eu me sinto até inútil, porque tudo que eu penso em fazer, poxa, ela diz, não, eu já fiz. Então, é um prazer muito grande. Eu falo em nome do laboratório, do próprio departamento de gastroenterologia, sabe? Você tem dado um exemplo muito grande para todos nós. Eu tenho um grande orgulho de colaborar com vocês nessas empreitadas e lhe agradeço até pelo seu exemplo de excelência, né, de trazer para nós esse workshop que é muito importante. E queria deixar aqui o meu agradecimento ao departamento. Falo em nome do professor Carneiro, que é o chefe do departamento atual, e queria dizer que temos muito orgulho do seu trabalho, né? Bom, vamos falar agora, depois dessa radiação de seda, né, professora? Mas com muito merecimento. Você merece muito isso, porque você foi quem criou esse workshop, idealizou, traz todos os, praticamente todos os participantes aqui, que são colaboradores dela. Então, é muito gratificante a gente estar participando e estar iniciando novamente esse workshop, que tem o objetivo de trazer as novidades, as inovações nessa área, né? Nós temos grande interesse de trazer o que está acontecendo de mais importante, né? Os novos conhecimentos, né? Na nossa área tem saído muita coisa. O transplante de células é uma área ainda inicial em termos clínicos, mas tem muita coisa já pronta, praticamente pronta para ser utilizada nos programas clínicos. E é um procedimento que vai simplificar muito desses procedimentos de transplante, né, que são procedimentos geralmente muito complexos, que envolvem cirurgias complexas. O transplante de células vem para simplificar. Aliás, a terapia celular é uma terapia que vem para simplificar os tratamentos, né, e torná-los mais efetivos e menos agressivos. É por isso que a gente tem grande interesse nessa área, né, e estamos trazendo isso aqui para vocês. O intuito desse workshop é ser bastante interativo. Então, gostaríamos que vocês participassem conosco, né, perguntem mesmo, essa interação é muito importante. Na verdade, eu confesso que não sou um especialista nessa área, eu sou cirurgião, e às vezes tenho até dificuldade de entender um pouco, né, a parte mais biomolecular, inclusive, mas sou entusiasta de tentar aprender, então todos nós estamos aprendendo, então sintam-se à vontade para questionar, nós temos um espaço destinado a isso no final de cada período, para que a gente faça esse debate, até para nós melhorarmos esse workshop, os próximos. No próximo, nós já vamos começar a planejar para tentar trazer para vocês também, de novo, o curso hands-on que nós fizemos, que foi muito interessante, no nosso último workshop tivemos isso, né? E então é isso. Vamos, então, tentar aprender o máximo possível, debatendo, e espero que vocês gostem do nosso workshop.
Obrigada, Flávio. E aqui é só para vocês terem uma ideia que, na verdade, um trabalho desses precisa ser multidisciplinar, porque se não tivéssemos a ajuda do Flávio, a gente não teria acesso ao material biológico, né? Aos fígados os tecidos que a gente precisaria para fazer esse trabalho, né? Nós temos a Márcia Kubrusli, que é do laboratório, e ela também é do Comitê de Ética da CAPEPESC, e ela vem ajudando o André Barateiro no projeto, né? Escreveu o termo de consentimento, que essa parte é uma parte muito importante também do projeto. Não estão aqui, mas eu preciso mencionar também o pessoal da divisão de anatomia patológica. O professor Venâncio tem nos apoiando, a figura do Dr. André Hirayama e Dr. Evandro, que são os patologistas. Por quê? Porque a peça, quando sai, precisa primeiro ser avaliado pelos patologistas, porque primeiro é o paciente, né? Então, primeiro a gente tem que garantir tudo do paciente, e aí o que restar, o que for descartado, é que a gente vai utilizar. E isso, só quem pode dizer o que pode ser descartado é o patologista. Então, nós temos a colaboração dos patologistas nesse trabalho, tá? Então, para vocês entenderem a dimensão de quantos profissionais, de quantas disciplinas a gente precisa estar colaborando para fazer um trabalho desse tipo, né? E a doutora Gabriela, que eu queria chamar para conversar um pouquinho, que ela é do Centro de Inovação de Tecnologia do Instituto Central, porque é um dos nossos desejos, né, que a gente consiga desenvolver algum produto para depois de inovação. E assim, nós contamos com o apoio do CITIC para a realização desse evento, por isso que eu queria convidar a Gabriela.
Bom dia a todos, é uma honra estar aqui com a Suzane. A professora Suzane é uma dos membros mais ativos do Centro de Inovação do Instituto Central, tanto como pesquisadora inventora, como madrinha de muitos projetos. Então, não só quase que uma dívida, o CITIC está apoiando esse evento, e eu queria dizer que nós estamos lá no Instituto Central abertos para receber qualquer ideia e integrar com outras disciplinas, outras unidades, como poli, enfim. Então, o que a gente faz é apoiar os diversos modelos de desenvolvimento. Então, muito obrigada. Eu cheguei esbaforida. Queria dizer que eu encontrei o professor Venâncio sem saber a sala, como eu. Então, eu até mandei uma mensagem no grupo, que é a sala 2303, né? Então, desculpa Leda, prazer em gênero.
Então, pessoal, assim, quem são das outras instituições? Todas as grandes instituições têm seus centros de inovação tecnológica. Eu recomendo fortemente que vocês procurem os centros de inovação das suas instituições. Quem é aqui do complexo também, cada instituto tem o seu centro de inovação, para vocês conhecerem o trabalho, né, dos grupos de inovação, porque existem potenciais produtos que às vezes estão sendo não vistos, né? E que poderiam gerar produtos de inovações importantes para cada instituição, tá? E agora, por último, eu queria convidar a professora Leda, ela vai falar um pouquinho da Emory e apresentar a Natália.
Bom dia a todos, é uma honra estar aqui com vocês hoje e participando pela terceira vez desse workshop. Nossa, é uma honra estar aqui com a Suzane e eu gostaria de lembrar vocês, eu estou na Emory, nessa universidade em Atlanta, na Geórgia. Vai fazer 23 anos já, e desde antes disso, Suzane e eu colaboramos muito com o meu chefe, que é da Emory também, na área de antivirais e em avaliação in vitro, e a gente tem um programa que, na verdade, a Suzane pesquisou bastante, que é o Sprint, que é uma colaboração da Emory com a universidade aqui, né, da Faculdade de Medicina, e iniciou por causa dela, e isso é muito importante, porque a gente consegue receber, a gente já recebeu duas pessoas através da Suzane, que foram e ficaram, uma ficou 6 meses, a outra ficou 4 meses, e a Suzane mesmo ficou 4 meses com a gente, aprendendo técnicas novas e desenvolvendo bastante coisa e aplicando aqui depois. E esse programa é bem legal. É uma maneira de incentivar, de pessoas irem para lá e virem para cá também, e colaborar com outros colaboradores da faculdade. E muito obrigada. Isso é muito importante e a gente quer continuar sempre com esse programa. Vai fazer o quê? 4 anos já que a gente está nesse sprint. É uma maneira de incentivar, e as pessoas têm que procurar isso e preencher formulários e aplicar. A gente não pode deixar isso morrer de jeito nenhum. E a gente aceita pessoas indo para lá sempre. É bem importante. A gente gosta de colaborar bastante. E muito obrigada. Agora eu vou fazer a apresentação, faltam 7 minutos. E vou começar, começar a falar em inglês? Vou fazer a apresentação da Natália em inglês, então. Natália, doutora Natália Carvalho Pellison. She's a product specialist from Grainer Bio 1. She has a degree in biological sciences from the University of São Paulo and a master's degree in cellular and molecular biology from the Faculty of Medicine of Ribeirão Preto at the University of São Paulo. She has extensive experience with cell culture and has participated in research projects to investigate the role of innate immunity in pathogenic protozoan infections. During her master's degree, she performed a high-throughput drug test for leishmaniasis. Since the end of 2019, she has been part of the Grainer Bio 1 team in Brazil as a product specialist, being responsible for the technical and scientific support of bioscience products such as magnetic 3D cell culture. And welcome.
Thank you. Thank you, Leda. Thank you, Suzane, and all the coordinators for the invite. It's a pleasure to be here and to present to you a little bit of 3D cell culture. Just a moment, please. Here is it. So we have 30 minutes to talk about a very huge content. So I'm going through some of the 3D platforms available for 3D cell culture. And I'm also talk about some applications that can be done using this methodology and some examples of cell lines that had already been used for this.
Tatiana, dá mais um clique na apresentação no rodapé aí para ela poder passar o slide.
Thank you. So I think it's very important to start from the beginning, right? To define what is 3D cell culture and why it's so important to have this method of culturing. So it's a very precious tool. So it's important for using for both developmental, cell, and cancer biology. And it's important because it brings realistic models. It is very similar or brings some characteristics to the culture that can be found in vivo models. So you have more relevant responses when you use it. It's important for mechanisms of tissue homeostasis, for accelerated translational research, and also for studying and understanding cancer biology. You may know or not, if you're not used to work with 3D cell culture, that we can find two different definitions for 3D cell culture types. You can find spheroids and also organoids. The definitions can be different depending the authors in the literature, but for having an idea about it, the differences, we can say that spheroids are like cellular aggregates that comprises a single cell type, or you can use also co-culture. And they lack the organized structures and specific cell types found in organs. So they are more simple than the organoids. On the other hand, you have the organized, which are self-organized 3D cell cultures that are derived from primary tissue, embryonic stem cells, or induced pluripotent stem cells. So they have similar functionalities as the tissue from which the cells originate. So it brings more complexity to the model, to your samples, if you use the most cells, the most components you have in the natural tissue. But why to do 3D cell culture? Why it's important to use 3D cell culture? I think the value of this model is that they represent more accurately the in vivo biology, and you can do this in vitro. So you can use this in your lab without the needing of using animals, for example. If we compare, 1 minute. I think we have some problem with the presentation. Okay? Okay, thank you. No problem. If we think about the methodology we usually use on the lab, we culture our cells in a flat surface, and the cells are removed from a natural tissue, and they go to a flat platform, a flat surface with a different charge from a plastic. And this model lacks a proper environment context. So you lose some important characteristics that cells find in natural. So you lose some of the structural architecture from the cells and this tissue, and this changes also some cells' functions. But when you go to the 3D cell culture, then you can replicate some critical features of it, and you have some morphological and also differentiation, polarity, proliferation rate, gene expression present there. here are some examples of the differences between these two methodologies. You can find differences, as I told you before, on the support, on the way the cells can be adhered. So if you think about it, a monolayer, they are adhered to the plastic. But then if you go to 3D cell culture, you have the presence of cell-to-cell interactions and cell-to extracellular matrix interactions, and this is very important for cells to realize their important functions. Also, we can see the presence of cell junctions and differentiation. They can be different in both models. And also, very important is to talk about the treatment sensitivity. So when you have monolayers, all the cells are exposed at the same conditions, the same quantity, concentration of treatments, but in 3D cell culture, then you are going to have different layers of the cells, and this exposure will be different too, and the responses to the treatment as well. So we talk about monolayers, cell culture in lab, and then we are talking about representative methods of in vivo conditions, right? Then we think about animals, using animals and animal models. But there are also some problems using them. One of them is the costs of using. You need to have a facility and people to work with the animals, to take care of them, to change all the needs they have. So this is a very costly model too. And also, I think the most important are the ethical issues involved in this. So we are thinking about animals and lives. And if we can find a different way to do our experiments, our samples, without the need of using animals, then it's better. You can reduce the number of animals, you can optimize your experiments too. And the last one, I think, is the interspecies differences. So sometimes we are studying human diseases, human conditions, and we are going to study in animals, which are good. They bring us some advantages too, but they are different species. So we can have some difference in the results as well. And thinking about treatments and development of some new drugs, it's very important to think that a very successful drug discovery campaign begins with the right assay. So an assay that measures biological process in a physiologically relevant and robust manner. So it's important to start since the beginning with a very representative methodology. You are going to have much, much better results at the end. When we think in the development process, we go through long years of this. And we start with target discovery and validation. It takes about 1 or 2 years. It can be more. Then you go to compound screening. You find different hits, leads to test. And then you find which one is working. Then you go to preclinical trials. And this takes a while, a lot of animals too. Then to clinical trials, where we lost most of the leads from the beginning. And at the end, you have some treatment available if you are lucky. And well, we start with a huge number of compounds, and then we want to at least have one at the end. But what if I tell you that you can improve this process and bring better results since the beginning, not having to wait until clinical trials to discover that it doesn't work? So the methodology used today begins with 2D cell culture. We start in the lab, validating our compounds, and then we go to animal tests, and then clinical trial, and the final treatment. But if you use 3D cell culture, or you incorporate it in the process, then you can filter better. You can pick better your leads or your results, and then you reduce the number of animals being used for it. We are talking about 3D cell culture and the importance of using it. So when we look at the number of publications, scientific literature available with these themes, this methodology, you can see the increasing of it, the number of it, in the last 20 years, at least. So it's very important. We know that every year, most people adopt the 3D cell culture methodology in their experiments because it's relevant, it's important. And also financial supports are asking for it to have this in your projects too. Let's understand some of the characteristics that are important and present in a spheroid or an organoid that makes this important for using. We have extracellular matrix presence, cellular interactions, concentration gradients, and gene expression. So the extracellular matrix is a very important compound. It brings not only structural, but also biochemical supports to the cells and the tissues. They are present in our tissues, right? In vivo tissues. So if you bring this to your lab, then you are going to be a very significant cellular interaction with it, with the compounds, the pool of growth factors that can be found also in there. So when you analyze tissues, different tissues has different compounds in their extracellular matrix. So they are very specific. The more you have these compounds, this present, the better are your results too. And if you change these compounds, then you will change and regulate cell signaling, proliferation, differentiation, and survival and adhesion. And also, extracellular matrix have important matrix stiffness. So this is really related to drug resistance in some models. So when we think about cancer models, for example, this presence is very important, and also for treatment discovery. So the most you can reproduce it in your lab, the better it is. Also, there are the cellular interactions present. So cells are interacting between each other in a three-dimensional organization. So when we think in a flat cell culture, they are only interacting on their sides. But here we have a three-dimensional interaction and the presence of adhesion molecules and other junctions molecules that are very important also for cellular signaling and activities. When we compare, and I like very much this picture because it's very visual, when we compare the presence of N-cadherin in this model, which is a glioblastoma model, we can compare the adhesion presence here and the distribution of cells as well. And we are comparing in vivo model with a two-dimensional cell culture. And we can see the difference here. So we have much more presence of N-cadherin here compared to the cell culture. But when we go to three-dimensional cell culture, then you can see they are very similar. They're almost the same. So this is why I say we have this representative model here. The other one is the concentration gradients, which are very important because cells are organized in different layers when we think about 3D cell culture. So the peripheral cells are exposed to high rates of oxygens, nutrients, and this makes them able to proliferate more. But if you go to the inside of the model, the sample, then you are going to find low rates of the same compounds. And this brings and represents a more necrotic or apoptotic region of the spheroids. And this could be important depending the model you are using. For cancer, for example, this is very important to have this hypoxic region on your samples. And we have to think about it because it's very different when we think about monolayers too. So the responses are very different in this case. So all this compounds the difference in gene expression levels. This is only for example, it's not a real result, for example, but we can see that spheroids are much more like in vivo in gene expression than they are with monolayers, because they are interacting, we have the presence of extracellular matrix. And these are very important when we are going to validate our models to evaluate if they are being expressed, if they are present there. This work, for example, they compared the in vivo with the, this one is adipocytes. So they compared white adipocytes extracted from mouse with some spheroids formed with the cells. And they saw that by mass spectrometry that most of the proteins found in the model of spheroids were present also in the tissue. Of course, we don't have all of them because tissues are more complex. We have different compounds. We can assure that it will be the same, but most of the proteins were present also in the in vivo tissue. This is very important. We can do this with monolayers, and also the time the cells can be cultured in 3D, for example, for adipocytes is bigger than in monolayers, for example. Let's take a look on different methods that we have available for 3D cell culture. Okay, so it can be different, and some classifications also can differ from authors to authors. But if we think about it, we have monolayers as 2D cell culture we already use in our labs. And then we go to more complex structures using inserts, for example. So we can use as barrier models. And as 3D cell culture forming organoids or spheroids, then we have different methodologies, which can be scaffold-based, scaffold-free, and also we have some different specialized platforms. Of course, I can't talk all of them. We have more, and every time we have different methods coming. But you have an idea about the most common ones, probably. So barrier models are this different approach that use differentiated epithelial cell cultures. They are mostly used as barriers, so you can form epithelial models like skin in the inserts. Then you use some medias with differentiation components and molecules, and this will make you have at the end some tissue here exposed. You can do airlift culture and expose them to in their air liquid environment. So this is important for differentiating some cells, for example, in the airway cells. So we have different approaches that can be done using inserts, like migration studies, which are not very 3D cell culture, but if you think about co-culturing and also organotypic culture, then you can consider them as a 3D cell culture too. This is an example of the differentiation of skin using an insert. Then they are mostly used for cosmetics studies now and tests, cytotoxicology tests in the cosmetic industry, replacing animal use, for example. They are already approved for using here in Brazil too. So you take about 13 days of culture, and then you have the different layers of the skin without using animals. Let's take a look then in 3D cell culture models. We are going to start to the scaffold-based ones. The cells in these models grow in the presence of a physical support. So they ranging from simple mechanical structures to extracellular matrix-like matrices. Some examples of this are the hydrogels and also some synthetic, it can be natural or synthetic hydrogels. Then we have the scaffold-free, which are self-aggregation of cells without the use of substrates or synthesizing their own extracellular matrix to form three-dimensional structures. Then we have different methodologies like hanging drop, forced floating, micropattern plates, and magnetic 3D cell culture. The last one are the specialized platforms. They will bring more complexity to the structures you are going to form, sometimes bigger than the ones you can form using these others, or you can associate others to use them at the platforms too. So we have some different methodologies here, which are organ-on-a-chip and also 3D bioprinting, or also, I forgot to add here, the bioreactors too. Let's start with scaffold-based, which can be natural or synthetic hydrogels. So for this 3D cell culture, you are going to allow your cells to grow in a very rich environment with ECM proteins. So you are going to feed them with these proteins. And this can be natural or synthetic, as I told you, and it's very important because cells need this contact to grow and to develop in spheroids. This is an example, one of the most used, which are metrogel, for example. It is hydrogels that are networks, so the cells get trapped there, and they grow and form spheroids, small spheroids inside them. So it mimics the extracellular matrix. It can have some compounds that are natural from the spheroids or not. So you can give them different extracellular matrix only for allow them to form the spheroids. So this is a process you can see. You use the extracellular matrix compounds with the cells in suspension. Then you add them in a small drop and let them polymerize and incubate for the period of time that it's needing. Then the cells grow and forms the spheroids inside them. And then you have to remove or use for what you want to. This was an example of scaffold-based. Now let's take a look in the scaffold-free methodologies. So hanging drop is a very common usage too. I think it's the cheapest way probably to do 3D cell culture because it uses surface tension and gravitational force to form the spheroids. You are going to use small drops of medium with cells, and then you are going to leave them this way. And by gravitational force, the cells are going to aggregate and form the spheroids. It's some tricky to use hanging drop because it's easy to make them, but it's hard to handling them, okay? We are going to talk about it too. Forced floating is also very used. It uses surfaces that are treated for avoid cell adhesion. So they are called low attachment surface treatment, and cells are impaired to adhere. So they are stimulated to interact between each other and form the spheroids in this way. It's very common to use both round or conical flat bottom to form this. Conical flats, no, sorry, conical only. And this way, they are forced to aggregate too, and you can use centrifugation or not to form them. The microplates associates a little bit of the last one I told you. So they also use low attachment surface, but they are going to bring these small spaces to the spheroids to form. So it's a way to have more high throughput number of spheroids in one well, for example. It can depends, the format can depend from the producer, the industry, but the aim is the same, to form small spheroids in each well. And also, I think this is the last one for this scaffold 3 is magnetic 3D cell culture. I'm going to give an overview, but Glauco will talk about the technology better tomorrow. So it works with magnetizing cells. You first magnetize your cells at the flask or your suspension cells if you need to. And then you go through a cell repellent plate and incubate them using a magnet. So you can do this by levitation or bioprinting. So with the magnet, cells aggregate very quickly, and then they can interact and form extracellular matrix. So here, we are not adding external extracellular matrix, for example. There's a small video showing the process. I think we have enough time to this. So these are the components that are used to magnetic 3D cell culture, are the spheroid drives and also the cell repellent plates and nanoshuttles, which you can add to magnetize cells. So this is the first process, to magnetize them. Then the nanoparticles are going to attach electrostatically. We're going to remove, detach them from the flask and transfer them to a cell repellent plate with the drive, magnetic drive. So here are the magnetized cells being added to each well. All movement. And then they are incubated and form the spheroids in a few hours. You have your sample ready to use. Well, if you want to know better and more about the magnetic 3D culture, I would like to invite you to watch the Glauco Souza lecture we hold tomorrow at 10 a.m. And the last one are the specialized platforms, which can be microfluidics devices. It's very interesting to use them because they integrate different models, different tissues, and you can have a more complex and complete response to them, to your treatment tests, for example. There are also the bioprinting platform, which is assisted by technology of printing the cells with some extracellular matrix, and this allows you to form more complex structures too. And the last ones are the bioreactors, which are some specialized vessels. You can cultivate a large number of spheroids at once, for example. So you provide them controlled conditions, such as nutrient supply, oxygen, pH, and all of this is controlled by sensors, for example. But there are some challenges. We have an idea about different methodologies, and each of them have their limitations. So when you're going to choose which one to use, you have to take this into account. If you are going to have some handling problems or homogeneity or reproducibility, how reproducible are your samples be, is important for science, for example. If we're going to watch them, analyze them by microscopy, is it available? Is it possible, easy to do? And if you need a lot of samples, you need a high number of samples, for example. So HTS is important. Is this methodology available for doing this? Some applications and cell lines. We have a general classifications here. So tissue engineering, cancer biology, drug discovery are some examples. So tissue engineering, you vastly use stem cells for doing it and differentiate the cells in your tissue of interest. Then you can use for therapeutics, regenerative medicine, or drug discovery, for example. It's very used, and we can see a lot of papers available with this methodology application, sorry. Cancer biology also, you can study better the responses, the molecules, the characteristics present in cancers, because you can reproduce better the microenvironment. You can add both cancer cells and stromal cells in your models and see they interacting. And for drug discovery, then you have the presence of physical barriers, which can bring a better result about drug resistance, for example, and also for cancer therapies, high throughput assays. If you're thinking, how am I going to validate this methodology, you have to choose the right process and mode of analysis. And some of them are very common. You already use them to 2D, but then you have to think that probably you are going to do some modifications, optimizations on the protocol, because we are talking about a more complex structure, and we have extracellular matrix present there. And probably you're going to, if you're going to do flow cytometry, you have to deal with that too. So it is easy, it is possible, but you have to optimize. Okay, so this is something that people sometimes forget. You try to use the same protocol, but it won't work. Okay, thanks that. Cell lines, we have multiple. Here are some examples that have already been used. But if you look at the literature, you're going to find multiple papers with different cell types, different applications, different approaches, okay? So take a look if there is already used some of your cell of interests. If you are going to work with primary cells, which are sometimes difficult, take a look if you already have something ready on the literature to help you to start, for example. And each model, each methodology can form a different form of spheroids. You can expect to have also always round spheroids. It depends of the cell biology too, okay? So we can see here in different pictures, there are different forms of organoids and spheroids. That's it. I would like to thank you again for the presence, for the organization committee, for helping us, for the opportunity. It's a pleasure to be here. I'll be available if you have any questions now or later at the moment. So here's my contact. If someone needs something or wants to talk about 3D cell culture, it will be a pleasure. Thank you.
Obrigada, Natália. Vou passar para o Flávio para apresentar.
It's a pleasure to present our next speaker, Professor João Nuno Andrade Ferreira. I was quite impressed with his resume. Dr. João Ferreira did his graduation in at the University of Porto. He is currently the director of he is an associate professor at the Faculty of Dentistry at Chulalongkorn University. He's the director of the Avastar Biotechnologies for Oral Health and Healthy Longevity Research Unit at this university, and the head of the QDent Bioprint 3D Lab. Dr. Ferreira obtained his graduation in dental degree in in the Health Science Institute in Portugal, and migrated to US. He was awarded with a clinical fellow in the University of Minnesota, where he did his master's degree in the resident program in orofacial pain and oral medicine. He received his prestigious Portuguese government Ministry of Science and Technology Award for his project of oral biology at the School of Dentistry in the Chapel Hills. He was also awarded with an NIH fellowship in these institutional labs. He has many awards, many important awards all over the world. He is now working at this Chulalongkorn University, and he was awarded with several international US, Singapore, and Thailand award, including Association for Dental, Oral, and Craniofacial Research. And Dr. Ferreira has published nearly 50 peer-reviewed works on oral facial pain and oral medicine. And in spite of all these activities, scientific activities, he makes also humanitarian work, giving, advocating and helping groups in international level for patients with dry mouth and dry eyes in the world. So we are very proud to have you here, Dr. João, and thank you very much for participating in our workshop. Thank you.
Good morning, all. Good morning, Brazil. I would like first to give thanks to Dr. Suzanne Ono and the University of São Paulo School of Medicine for inviting me. My name is João Ferreira, and unfortunately, I'm not available to be online on April 11. So I decided to record this lecture. And the title of my lecture is In Vitro Modeling of Oral and Maxillofacial Tissues via 3D Bioprinting and Physiological Dynamic Systems. Again, my name is João Ferreira, and I'm Associate Professor at the Faculty of Dentistry, Chulalongkorn University, based in Bangkok, Thailand. And I'm the Director of Avatar Biotechnologies for Oral Health and Healthy Longevity Research Unit at Chulalongkorn University, and the head of the Student Bioprint 3D Lab. So an overview of my lecture today. First, I will be talking about the challenges in organoid biofabrication, and particularly focused on the salivary gland. Next, I will talk about some of the strategies to bioprint these organoids via magnetic nanoparticles. This technique is called 3D bioprinting or bioassembly, or M3DB. And we use in our lab these three techniques: the M3DB with bottom-up assembly, levitation air-liquid interface, and ring-based assembly. So I will be describing a little bit how we do this and some of the outcomes we got from our experiments. Next, I'll talk about Matrigel and basement membrane extract alternatives for organoid biofabrication, followed by the integration of different cues with organ ownership systems. And in particular, I'll show some data for the oral mucosa. And finally, I will talk, sum up, and we'll talk about future work. So why would we study salivary glands? So unfortunately, a lot of the patients that we see in the oral medicine clinic are head and neck cancer patients. And the majority of these have to undergo radiation therapy. Both partial RT or radiation damage or global RT damage have a severe impact on the secretory epithelium of the salivary gland, which is the gland that produces saliva, an important fluid to lubricate our mouth, to chew, to talk. And so this radiation therapy not only damages the secretory epithelial cells, as well as the nerves. And what types of treatment do we currently have to tackle this problem? So these patients, of course, have severe dry mouth, as you can see here in the left image. And because, of course, of the loss of secretory epithelia and hypofunction, this hypofunction can be, though, somewhat alleviated with current oral basic care products, such as oral rinses, moisturizing sprays, oral balance, and this is one of the popular brands out there, Biotene. Also, this can be alleviated using medications, and in particularly muscarinic agonists that act on muscarinic receptors M1 and M3. So in this particular case, we use pilocarpine or cevimeline. But medications that are cholinergic stimulants, they do have side effects, which can be cardiac. People may complain of sweating. And ultimately, the loss of salivary secretory epithelia will only provide to these patients a short-term relief. So we need to build new secretory epithelia, basically, as a new approach to tackle this problem. So how do we going to do this? So one way that has emerged to overcome the lack of epithelial tissues is by regenerating, right, secretory epithelia. And one way we have to do this is by creating what we call organ building blocks, or OBBs. So this concept has been out there, since 2009 or so. And this idea of creating these building blocks is very important because in vitro, we cannot really assemble very large tissues. And these organoid building blocks, when we're talking about organoid building blocks, we're talking about organoids, we're talking about spheroids and assembloids. So basically, 3D cell constructs that one can assemble via multiple techniques. And these organoid building blocks, or OBBs, will have ultimately applications for regenerative medicine, but also other applications, personalized medicine, in particular drug screening, infectious disease modeling, drug discovery, right? So, and this organoid effort in this OBB era has come to light, and it's quite hot at the moment since the United States Federal Drug Administration has enacted its 2.0 Modernization Act, which allows faster and most cost-effective drug development via in vitro physiological systems, such as organoids. What are the key factors in organoid biofabrication? So one has to take into account that in order for us to assemble an organoid in vitro, we need to select the right cell source. We need to select the suitable factors that will drive those cells to differentiate and produce ECM and be functional. We may need a matrix to support those cells. Physical cues, which are important to tune the microenvironment that we need, in particular if we're using stem cells. And finally, we may also have integrating cues to have some physiological microenvironment that is similar to the organ being studied. And these integrating cues can be what we call OOCs, or organoid on chip, or multiple organoid on chip systems. And these allow for the recapitulation of the physiology in vitro. What has been done for the salivary gland in terms of organoid biofabrication? So everything started back in 2010 or so by a group, my colleagues in the Netherlands, led by Dr. Rob Kopis. And what they did is they created this in vitro floating clusters, okay, also which they also called salispheres. And these salispheres were the first organoids in the salivary gland field to be created. These were basically aggregates of cells, of primary cells, that were initially taken from the mouse, and then these were reproduced with human primary salivary gland cells. And these cells were embedded in matrigel, which is an animal-derived extracellular matrix, to recapitulate the morphogenesis of the salivary gland. That's the main purpose. And as you can see here in A, you see a salivary gland branching. And ultimately, what they found too is that they can rescue function upon transplantation in mice. And in particular, they use different types of sorted cells. One of them was CK positive cells. One of the critical issues here is not only the fact that they use matrigel, but also the presence of certain stem cells do decrease as we age, okay? And they become very, very scarce. So that is, of course, a problem. And therefore, some of these initial salispheres included, which included CK, now actually include all types of different cells. And so heterogeneity is important in these organoids, as well as the ability to proliferate, because ultimately they need to be transplanted into the human body. Another beautiful organoid platform has been published recently, more recently, by my colleagues from Yonsei University in South Korea. The main investigator in this effort is Professor Jae-Yo Lim. And what he did, established functional human salivary gland organoids as well as tumoroids, so cancer derived salivary gland organoids, using different media conditions, what they call GM and DEM. So this process takes over 2 weeks and basically involves both a mix of different cocktails, different medias, and I will not going to go too much into detail, but they also used growth factor reduced matrigel in these systems, which of course has limited translation applications. And they used a vast array of growth factors. So, as you can see, some of the challenges when we fabricate organoids in vitro are, as I mentioned, the use of ill-defined matrices like Matrigel, but other issues can arise. Accessibility to the biopsies, availability of the cells from donors, the lack of maturation of the cells in the organoids, lack of heterogeneity, the misrepresentation of the native organ in these organoids. Also, there's lack of cellular microenvironment that we see often. And there's a huge lack of multi-organ studies. Functional status assessments are limited in organoids, and the clinical grade process for these organoids sometimes can be slow to set, since a lot of us are using different reagents from that are not clinically great, and therefore a huge process of translation of what we do is what will take place ultimately. So I'm going to focus on three things that our lab decided to tackle. One of them is to replace some of the ill-defined matrices that are animal-derived, Matrigel, basement membrane extract, but also looking at also how the cellular environment is important and how functionality is critical, particularly when we are trying to recapitulate the secretion of the salivary glands. So we used an approach called magnetic bioassembly or bioprinting, or M3DB. And the idea behind this is that M3DB is important to spatially organize, okay, the cells in 3D, and it's highly reproducible, and ultimately what we want to achieve is the generation of micromodular compartments and allow the cells to endogenously deposit or produce the extracellular matrix, and ideally with a free necrotic form. So this fabrication of micromodular compartments is also called microtissue engineering. So ultimately, we don't want to have that necrotic form, but this depends a lot on the cell density that we have at the initial stages of our cell culture. This magnetic 3D bioassembly approach was possible because of our collaboration with Dr. Glauco Souza, which at that time was not part of Brian or BioOne. He had his own startup company in the United States, and we did some work together with these magnetic nanoparticles, which are called nanoshuttle. These magnetic nanoparticles, and he will mention that in a later talk on April 11, these magnetic nanoparticles are basically particles that are composed of polyalysine, gold, and iron oxide. And the beauty of these is that the polyalysine component allows one to use these magnetic nanoparticles and tag the cells by electrostatic interactions. And this is possible after you culture your cells in a 2D, in monolayer, in a flask. You have your magnetic nanoparticles. You will wait for a few hours, usually 3 hours, and then you can detach the cell using any cell dissociation reagent. And after that, you have multiple options. Either you place the magnets on the top to levitate the cells in 3D, or you place the magnet at the bottom of your plate. And these approaches, we call them the M3DL and the M3D approach. So the bottom-up approach, M3DB, at the air liquid interface, the M3DL, where basically your 3D cell cluster is going to levitate. So our first question that one of my PhD students at the time did was, where do these magnetic nanoparticles go? So we saw that these magnetic nanoparticles do tag the cell membrane initially, and after about a week, they are released into the extracellular matrix, and eventually, after a month or so, we have a very small amount of magnetic nanoparticles still in the extracellular space. So these magnetic nanoparticles, as I mentioned, they tag cells through electrostatic interactions. And of course, if you replace the media very often, they are quickly washed out. So the first approach we had to develop our salivary gland organoids was to bioprint the organoids using a bottom-up approach and by placing these magnetic dot, or also called magnetic pin, under the 96 well plate. So we did this first approach using adult stem cells that are neurocrest derived, important in the development, differentiation of the oral primitive ectoderm. These cells can be collected from the human dental pulp, which is the tissue that is inside your teeth. So basically, so pulp of extracted wisdom teeth, okay, can be retrieved, and you can collect these cells. These cells are called human dental pulp stem cells, and one can produce spheroids. Okay, so these sphere-like clusters using the M3DB, and one can look at the protein expression of these cells and cell constructs. And what we found was that these spheroids can be assembled very quickly, within 24 hours, literally. And we can maintain both mitosis, multipotency, as well as neuroectoderm and epithelial markers. We're talking about markers such as PITX1, 2,3 or beta 3 tubulin, and also epithelial markers, keratin 14, keratin 19. Then we, in the second stage of this process, we added retinoic acid and fibroblast growth factor 10. And this is for inducing the differentiation of these neuroactive cells, or neurocrest-derived cells, into the epithelial glandular stage. And so one way to basically check whether these cells have epithelial function is to check or assess alpha amylase activity. And when we increased the amount of FGF10 that we had in the media to 40 or 400 nanograms per ml, we could see that alpha amylase activity to increase. So alpha amylase is an enzyme that is widely abundant in human saliva. So we also checked epithelial genetic markers, keratin 14, aquaporin 1, mucin 7, and amylase 1. And we confirmed that when we have the FGF10, we can induce differentiation. Now, this is based also, the idea of having FGF10 was based basically on knockout mouse studies where they knock out FGF10. And those knockout studies show that the salivary gland does not develop when we knock out the gene for FGF10. So, so we were able to induce the differentiation. We could see alpha amylase activity and pro-actin markers. Plus, when we added cholinergic stimulants and adrenergic stimulants, so in this case, we added carvacol or CCH and isoproterenol, we could see a peak in calcium activity, as you can see here in real-time videos. But also we could see salivary gland epithelial tight junctions in transmission electron microscopy, and also TER, which stands for transepithelial electrical resistance, which we found that in our salivary gland differentiated spheroids in the black columns, we saw those that resistance, that epithelial resistance, boost up when compared to undifferentiated spheroids. Then we wanted to ask the question, can we actually induce the regeneration of salivary gland epithelia ex vivo after radiation? So we irradiated fetal salivary glands ex vivo, and we implanted our salivary gland organoid. And what we immediately found was we found this sprouting of innervation from the organoid to the fetal gland. And the other thing that we also found was promotion of mitosis, both on the irradiated gland and in the organoid. So the irradiated control, as you can see, has very little mitosis. And this was very striking to see this rescue of mitosis, this promotion of innervation. We found this in the radiated gland, but also on the non-irradiated gland. So, and you can see the post-ganglionic neuronal fibers spreading from the salivary gland organoid to the fetal gland. We could measure epithelial growth in this ex vivo culture. We can measure only up to 72 hours or 3 days. We found that the implantation of the organoid in this ex vivo fetal mouse clearly improves the salivary gland epithelial growth. One of the major issues, although with these organoids, was that they were actually quite large. They had about 1 millimeter in size, close to 1 millimeter in size in terms of diameter. So maintaining its central core viability was an issue, and was very difficult to maintain the viability of 80%. So at least. So what we then try is we try to use smaller spheroids, and we wanted to test if we could develop high heterogeneity cancer organoids from salivary gland cell lines. So we use this HTV41 cell line from ATCC, a repository in the United States. So this is a mucoepidermoid cell line. And what we found after we stained with Ki67 nicodierin, which is an epithelial marker, Ki67 is a proliferation marker, as I mentioned before, we found that when we assemble these spheroids with nanoshuttle magnetic nanoparticles in the bottom-up approach with magnetic field under the plate, we're able to see this beautiful mitosis all over the organoid. And the organoid is very consistent, sphere-like morphology. When we try to do this using a self-aggregation technique without magnetic nanoparticles, the story is very different. We see a lot less heterogeneity, a lot less mitosis, and the reproducibility, particularly that sphere-like morphology is somewhat lost, okay? And this may relate to how these spheroids are manipulated ultimately. So the user has to have enough practice in order to manipulate these spheroids when nanoshuttle magnetic nanoparticles are not used. But strikingly, one has to think about whether heterogeneity is present when we want to develop cancer spheroids, and in particular salivary gland cancer spheroids. So this is an ongoing work. Hopefully we're able to publish this very soon, and I'll be able to present this at a certain point. So the other technique we use is the magnetic 3D levitation. And this is based on systems where we place the magnet pin or a disc on top of the plate. And the air media interface allows you to actually create larger tissues than the bottom-up approach. So because tissues can be reperfused with air that and the oxygen you have basically in your incubator where your cells are being cultured. So we use for this strategy, we use primary cells from the salivary glands of pigs, which we can have access to. For us, it is a little bit more challenging to have access to large amounts of human salivary gland cells. So we use this as a proof of concept, these porcine salivary gland cells. We're able to assemble them really, really quickly as these spheres in the air-liquid interface. And the viability, as you can see here, after 5 days is, is still quite high, 50 to 75%. So, but with time, this viability decreases, the viability of these magnetic biassembled spheroids at the air-liquid interface. We wanted to look also at the presence of innervation and function in these levitated salivary gland organoids. We found that there are cholinergic markers present, there are panorrhonal markers like beta-3 tubulin, and after we use a calcium-based assay, we found that when we have carbachol, which again is a cholinergic stimulant, we found an increase in calcium activity, and we did all the fluorescence studies and saw that calcium influx and mobilization by the cells in these levitated organoids. We also look at alpha amylase activities, again, as a marker for function of the salivary gland. So we found that stimulating the gland with carbachol, with a muscarinic agent, can increase alpha amylase. Next, we also explore an approach that allows us to create ring shape or ring-like structures, and we found this to be quite useful for our ductal salivary gland cancers, and this can be achieved in only 3 days, so quite quickly. Quite quickly, we can assemble basically these, I call this and joke with my staff that we should call these crispy donuts because they are really, really look like donuts, in particular here in the confocal images. So, but anyway, these can just follow the normal protocol for magnetic bioassembly, which is you first type the cells in monolayer culture in 2D, then you dissociate the cells after 3 hours, then you bioassemble in 3D using that ring-shaped magnet at the bottom of your plate, and overnight you have the formation of the ring. At 48 hours you can see this very nice ring, so after 48 hours. So you can then, after 48 hours, you can then model this ring the way you want, either with radiation or chemotherapy, and that's what we're currently doing. Now you see a lot of peak adherence, so and cell adherence junctions, and that's really key in putting together this salivary gland cancer or ductal cancer. So then I would like also to show you an approach where basically we started this during COVID when we found that we could not have access to matrigel. So we decided to move on and fabricate our own decellularized extracellular matrix. So we used the pig salivary gland tissue to do so, specifically the submandibular gland of the pig. So we used basic anionic detergents, SDS, endonucleases to remove the DNA in this process. We studied particularly 0.1% SDS and 1% SDS to remove all the DNA content. We were very successful on that. On that end, as you can see here, there's no DNA content. We also quantified the DNA. We found less than 50 nanograms of DNA in dry ECM, decellularized ECM tissue. And then we culture cells. We culture primary cells. We culture also cancer cells. We found with primary cells that we can have a very nice proliferation profile with 0.1 and 1% SDS, and in particular 1% SDS actually promoted quite strongly the proliferation of primary cells from the salivary gland. And these was quite striking when compared to your BME, based on extract, which is similar to matrigel here, these black columns. So after 3, 5, 7 days, our 1% SDS produced submandibular gland ECM was strikingly better in terms of proliferation viability to generate these salivary gland organoids from primary cells. So we also looked into protein expression. We found that the DCM can enrich in particularly acinar or pro-acinar cells, particularly the aquaporin 5. In terms when compared to BME here in red, and this is quite strikingly here with 0.1% SDS. For ductal and myoepithelial markers such as keratin 19, alpha smooth muscle actin, we did not see major differences between the BME and our decellularized ECM from pig. We also looked at cell adhesion, cell tethering via SEM, and we found that the cells really attach quite well to the DCM. And therefore, we plan to use these decellularized ECM for future studies, and particularly to integrate these with organ-on-chip systems. Talking about organ ownership systems, so those systems, as I mentioned here, are important to integrate the cues, key physiological and structural features of your native organ need to be placed into these in vitro systems and considered as to mimic or recapitulate the native organ, physiology, and function. So one can use single OOC platforms or multiple OOC platforms. Here you can see how big this field has grown. You have here the number of collaborative studies from industry looking at different OOC systems from different organs, and in particularly the gut, liver, heart, kidney, neurons, so lungs. So these are the most popular organ models, as you see here. And there are already several commercial companies making this organ ownership system. One of the most dynamic one is MyMedis, based in the Netherlands. So we have done a collaboration with a company based in Italy in order to see whether these OOC systems can improve epithelial mitosis and cell viability. So what we see is this system is basically here displayed on the left upper corner, and what you have is you have these chambers placed on a platform, and they're connecting with the silicone tubing with a peristaltic pump inside the incubator. And this single organ platform have basically a dynamic flow and does use a considerable amount of media. So that's one of the limitations of these platforms, of course. So so you can reperfuse these organoids by using these transvaal inserts in these chambers, and there will be media flowing underneath. Comparing static with these dynamic systems, cell viability improves substantially from 85 to 95% when we develop oral epithelia, okay, using the magnetic bioassemble systems. This can be done at the air-liquid interface. One can put these basically oral epithelial cell sheets into the transvaal and look at different markers. We've looked in these preliminary studies, we looked at proliferative markers, so Ki67, but also epithelial markers such as hicadurine, but also tight junctions such as onoculins 1. We found that when we place these oral epithelial cell sheets into the dynamic flow systems, and in particular the MIVO, we saw this increase in cell proliferation, but also the proliferation of other markers. We've assessed this using not only immunofluorescence but also Western blot. And in confocal, we see an increase in epithelial markers, both hicadurine and tight junctions. So as a take-home message, I would like to wrap up and say that the bioassembly platforms using magnetic nanoparticles, both M3DL and M3DB, can generate viable and functional salivary gland like organoids. And the necrotic core is minimized, but if one decides to seed a lot of cells, and we did that on our earlier studies, we may have a problem. And therefore, I recommend you keep your spheroids in the system at 300 micron or below. in terms of the diameter size. So if you want to use, or if you want to save more cells into your system and develop larger organoids greater than 300 micron, then we recommend you use the levitation systems or fluid flow systems, like the organoid on chip with constant media reperfusion. The bioprinted organoids, we found that they can support salivary gland regeneration, and we also found that their EVs are key here. I didn't have the time to show that, but we did find that the extracellular vesicles are important in these ex vivo models. Organoids from M3DL and Hen3DB can be integrated in fluid flow microphysiological platforms. And I showed you some of those early studies. Magnetic bioassembly platforms can support the ECM substrates if decellularized ECM is needed to replace animal-derived ECM, for like BME or Matrigel. And we found that also that millifluidic OSC improve oral epithelial cell mitosis and viability, in particular in our oral epithelial or oral mucosa models. What we're looking for next is to integrate with fluid flow OOC systems for organoid in vivo transplantation, also disease modeling. We want to model different disease for salivary glands. We want to model radiation damage. We want to model chemotherapy damage. We also want to screen for new drugs or repurpose old drugs, and investigate also the tumor microenvironment, particularly the salivary gland cancer organs. So we want to do this with millifluidic system, but also with microphysiological systems that minimize the amount of media that you use. But this is possible, all this work, because I have a fantastic team, very diverse team of individuals from all parts of Southeast Asia. So the work that I showed you was possible because of Krista Baladin work. She's from Indonesia. She's now based in Singapore. Ajima Shantaroj, she did the EV studies, so she was my postdoc. She's from Thailand. Khurshid Ahmed is from Pakistan. So he did the work with the decellularized ECM. And then I have many graduate students here that did some of the work with the oral mucosas, such as Tuan, Van Van, and Kientron. So I'm very thankful for their work, their dedication. And of course, I would like to give thanks to my funding agencies, the National Research Council of Thailand, Chulalongkorn University funding different funding programs, but also the International Association for Dental Research, and in particular GSK. Greiner Bio1 also supported the use of the magnetic nanoparticles. For the millifluidic system, we were able to collaborate with React for Life with the biomedical engineer, Professor Silvia Scaglioni. And other works are being continuously developed with other collaborators from startups in Chulalongkorn, also from the collaborators from the National University of Singapore and Mahidol University. So thank you very much for your time. This is my email if you want to contact me if you have any questions about my research. Hope you enjoyed this talk. Thank you. Hope to see you one of these days.
Vamos fazer agora um break, pessoal, de meia hora. Vamos voltar às 10h45, tá? Nós temos aqui embaixo, no térreo, um local onde pode ser comprado algumas coisas se vocês quiserem, tá? Nós encontramos, então, 15 para as 11. Até mais.
So let's continue. It's a great pleasure to introduce Professor Leda Bassiti. She's an assistant professor at Laboratory of Biochemical Pharmacology at Emory University. And she possesses extensive know-how focused upon the studies of data analysis, in particular regarding drug resistance, mutations, and profiles in HBV and other relevant virus pathogens. And she has 28 years of experience in virology and viral diagnosis, working with HIV, hepatitis B and C, West Nile, dengue, chikungunya, yellow fever, and Zika virus. So, thank you, Professor Leda.
Thank you, Marcia. Thank you for inviting me to participate in the workshop. So we are going to talk about the 3D cell culture application for drug discovery. And most of the 3D cell culture that I did in the lab was with a grainer product that is called the Nano Shuttle. And we used for many viruses, including Zika. We used for yellow fever, myrovirus. We used this system for brain tissue, and I'm going to show also some work that we did with the hepatitis B virus and using adherent cells. And our goal for hepatitis is to culture the cells, long culture, let them stay for at least 1 month being cultured in the in vitro, so we can detect cccDNA, which is a little difficult because it's just a very low concentration of cccDNA, and it takes a while to be produced. And our goal for hepatitis B virus is to develop drugs that inhibit cccDNA production and replication, and that's what we do in the lab related to hepatitis B. And this system is really nice because of the possibility of culturing the cells for a long time. So here it's this one. Which one? So what I'm gonna show, I'm gonna start the talk about showing some pictures of the spheroids. So here we call organoids, but as Natalia said, you can call them spheroids, organoids. But let's see now if I can. Maybe she can. Huh? Foi? Foi. Right. Okay. So, so here I think this slide was already presented, so I'm gonna go quickly here, but that's the system that, okay, thank you. That's the system that we have been using in the lab. So, and the advantages of using this is because, as Natalia said, it mimics native tissue environment, is a fast 3D organoid assembly. So the matrigel is also very good, but it takes a while, about 3 weeks, to make the organoids. Instead, this one, it takes 15 minutes to make the organoids. So it's incredibly fast, especially when you are using adherent cells, but also with primary cells, the system is really good. And here we use the bioprinting system, not the levitation. Most of the time we using 96 well plates. And as she said, also there is a good maintenance of phenotype and function, which is important for us to keep the cells happy and have also cell-to-cell interactions here. So we also like the long-term culturing. As I said, we want to keep them as long as possible. And we love this to use this system in the future. We have not done yet, but we would like to do the drug resistance with the system. So you keep feeding the cells with drugs in the low concentration and keep them until you see mutations showing up. But for this, again, we need to keep them in culture for a long time. And also for antiviral toxicity and drug metabolism, and mainly for human primary hepatocytes. So here, our first questions when we started using this 3D system is, can 3D organoids improve the evaluation of antiviral agents for clinical use? And also the primary human 3D liver organoids, we would like to get the tissue and do some dissociations and add to the plate. When we do that for a 2D and monolayer, the cells are not really happy. But instead, when we use the 3D organoid system and then keep them interacting to each other, it's much better. So we also would like to see for several viruses that I cited before, as Zika virus, GV, GCV, Mayaro, chikungunya, yellow fever, HIV. We all use these viruses in the lab testing the antiviral drugs. So we started, why not, like thinking, why not to use murine brain organoids and infect them with make the organoids, brain organoids, and infect them with these viruses and look at the morphology, see, because some of these viruses are cytopathic. We want to, see the morphology of the cells in the format of organoids to look at what happened before we add the antiviral drugs. So here we started this study with brain tissues from mice. And we used also it's like a blender, but for tissue from this company called Mutiny Biotech. And it's a homogenizer. It's important to just take the tissue like the liver and then use a system like this, and it's called Octomax tissue homogenizer, and add the tissue there and prepare it for making the organoids. But this is really quick. So we did this, the single cell suspensions, and using enzymatic degradation, and then we, after we prepared the tissue, we did a cell counting to see how many cells per ml we had for the 2 brains that we used to see the viability of the cells before we made the organoids. And we had a good amount of cells there. And so we knew how much of the cells we're gonna do this, make these organoids.
Vai dar uma atualizada aí.
So then we prepared them and made like a scheme here, so to look at how we're gonna do this process and how many days we're gonna differentiate the cells and then infect them, and then look at by PCR the production of a virus in the supernatant or even the cells, and we in parallel use the Viro cells in a 2D system. But anyways, we used these viruses here and infected them for 6 hours, removed after 20 days of differentiation in the 3D model. And here I'm gonna show some pictures of day 1, day 8, and day 26, side by side with the 3D model, the spheroids that you can see here on this figure. And it's really quick that like day 1 already you see the beautiful spheroid, and then day 8 they compact a little better. The picture's not so great, but nevertheless you see the monolayer that the cells are not happy, they are not interacting with each other, especially like, after 8 days, 26 days, you see the difference here. And we liked these pictures, and then we infected them. And here are the name of the viruses that we used, like the GCV, GEV, the enterovirus 71, the Mayaro virus. My mentor he has a passion for Mayaro virus, so it's more common in Brazil than in the United States, but he likes studying this as well. So we have this virus in the lab trying to find drugs against that. So, and Zika at that time. So you see that some viruses here, a cytopathic effect, and you see the organoid being like a little bit, the morphology is clearly that showing that on GEV, for instance, that the the virus is causing a cytopathic effect. So you can see the morphology of that, and enterovirus as well, and JCV as well. Well, we liked this. So then we took some examples here for the doing a PCR. We did an extraction from cells and also from production of virus in the supernatant, and look at CT values here. I'm saying it's crossing point, but it's like the CT as well. And the delta CT indicating that relative to the cell control, the lowest the CT value, more production of virus you have, intracellular and extracellular for the Mayaro virus, indicating that the virus is replicating here. It was a good example that the system is working very well, at least for this virus. And then we look at the Zika virus, especially in the brain. So again, we took this brain from mice and did the dissociation, and then all this work made the organoids, infected them, and removed the virus, washed the cells, and this all in organoid systems. And again, you see a good production of virus replication, intracellular and extracellular, seeing the lowest the CT value, more replication you have, more viruses here. And the system worked really well here. So we were happy with this. So this is an example, and then can be applied for what we have interest is in. It's organoids from liver, from the tissue, that we can do exactly the same. And it works really well. So this is an example of, I think Natalia said a little bit about this MPCC, the micropattern co-culture. Is also, it was used a lot. I myself used, and each plate costed, I guess, almost $5,000. And it's one-time use. This is the MPCC that you see, co-cultured with the fibroblast, because it's the primary hepatocytes, no? Co-culture. So the plate already comes with the cells already there for you to add the drugs and look at the infect the cells, and then add the drugs, and then look at the viral replication or activity of the antiviral drugs. But it's really hard. I lost my plates because it didn't work well. I could not have a good replication of the virus there. And I'm showing a 3D co-culture in parallel here for you to see how beautiful the organoid is in comparison to the MPCC after 11 days. And it's really hard to, just not to touch them, to disrupt them. And I don't know if you can see, but the fibroblasts are around that circle of primary hepatocytes. I'm like a little, can I touch here to show? So the primary hepatocytes are here, and the fibroblasts are here. And this is what we see the culture. Can you see the difference? This is all the primary hepatocytes. And they love the fibroblasts. So they need to be co-cultured to be happy there. And that's the system actually that it was developed by Charles Rice in his laboratory that won the Nobel Prize. So, but yeah, but it's really hard to use and expensive. And so here was a system that we used with the cells already transfected with the hepatitis B virus. It's called HBV-AD38. So they have already the virus integrated in the human genome, which is the hepatoma cells called HepG2 cells, and they are called HepAD38. And this is controlled by the antibiotic called tetracycline. If you add the tetracycline in the media, you stop production of virus. But if you remove the production of virus, and it's infectious. So you have to use in the BSL-2 laboratory these cells. So, but it's controlled by the tetracycline system, which is good for drug development. We use this system a lot in the lab in a 2D system, not a 3D. So as we don't have to infect these cells are good to do these kinds of experiments, making organoids and looking at co-culture with CD4, like I was trying to see if I could use 2 different viruses and look at maybe add drugs and see activity, if there was any antiviral activity for both HBV and HIV. And in this kind of hepatoma cells, the organoids are beautiful, huge and beautiful. And here I was doing monoculture and co-culture to get like in parallel to just to play with the system and look at the antiviral drug activity. And here I show an example of another system where we have to infect the cells with hepatitis B. That's called NTCP clone 7, which is these cells are HepG2 cells, but they have overexpression of a receptor for sodium bioacid co-transporter receptor that it facilitates the infection with hepatitis B. So you infect these cells with hepatitis B, you have a higher probability of the cells are being infected with that. So I used this because you can play also a little bit more with the system in terms of what kind of drugs you're gonna analyze. If it's gonna be an entry inhibitor, the system with HepAD38, you cannot, because the cells are already transfected with the virus. In this system, if you wanna use a new drug that inhibits entry of the virus, you can infect them, you can treat them first with the antiviral and then infect and see if the virus, if the drug inhibits entry of the virus. So, and here it's just an example of the co-culture that we did. And Suzanne and I are always looking at how can we improve a system to look at an easy way to look at the CCC DNA inhibition. But first we need to get the cccDNA production to be able to add a drug and say, oh, is it inhibiting cccDNA production? If we don't have a good level of cccDNA, we cannot say that the drug is active. No? So this system is again with the organoids that we were playing around in the lab to look at, can we detect cccDNA once we make these organoids and leave these cells like for a long period of time incubating. So we looked at and we saw that not that much production, but we still see production of cccDNA when we made these organoids and let them be incubated for a long time. We see a lot of HBV DNA, but we can see in 2D as well. But here we were, oh, this is really cool. We can see it as well. Unfortunately, I also used the co-culture here with CD4 and CD4 T cells and look at HIV RNA and could not see anything in this system. But I was happy already that we could see cccDNA here. And here is another example that we did with those cells that which we had to infect them with the hepatitis B virus that's called the NTCP clone 7. We did the same system here. And here we could see HIV, however, we could not see cccDNA. Perhaps the amount of virus that these cells were infected with was too low, but we were unable to see production of cccDNA here. Nevertheless, we were happy to see that the co-culture in 3D system, we could see HIV RNA here. And again, this was the PCR, the RTQ PCR that we did, and that's copies per ml here. So, and here we just played around, just showing the 96 well format, how you can, you know, just play with the system, make rapidly these organoids, and look at for several days incubation and harvest cells and soups. And we did with the HepAD38 system here, and showing again the organoids here, how good they are. And we looked at some of the drugs here. And that map that I showed with the HepAD38, we added some drugs after we made the organoids, and look at by PCR, the HIV RNA, HBV DNA, when we added these two potent drugs for inhibition of HBV DNA replication, mainly here, we could see that the drug at 1.1 and 0.1, 0.01 micromolar, you see a drug HBV DNA inhibition here. The highest the drug concentration, more inhibition you have, and then you calculate the EC50, EC90 here in micromolar, how much of this drug inhibit 50% of the replication, 90%. And we looked at just to see how good the system was when we made the organoids and added the drugs. And this was just an example to see that the system works well. And the conclusions are here that this system of co-culture organoids was created here in our lab, and for these, at least these 2 viruses that we played with, these cell cultures that we made, and it worked well. The immunofluorescence I'm not showing here because of time, but worked really well as well. And the NTCP, we could, again, I didn't show, but shows the production of P24 capsid protein in the supernatant. And, but most importantly, this model is validated and reproducible and provides multiple and simultaneous modalities to quantify HIV and HBV replication, and the impact of novel antiviral agents on desired events, especially for the future now that we know that works with brain tissue we can use for liver tissue and make organoids and study the cccDNA and other markers. And I would like to thank, we had several people from our lab that contributed to this work. And of course, the Grainer also helped us with all the reagents to make these organoids. And thank you, everyone. So I'm going to present Adam Gratz. Adam Gratz is an assistant professor in the Division of Digestive Diseases in the Department of Medicine at Emory University School of Medicine in Atlanta, Georgia. He's a member of the Cell and Molecular Biology Research Program at Winship Cancer Institute. He's also a member of the Biochemistry, Cell and Developmental Biology and Genetics and Molecular Biology graduate programs at Emory University. He's also he received his PhD and his Master of Science from University of North Carolina at Chapel Hill. He completed his postdoctoral training at University of Pennsylvania and at University of North Carolina at Chapel Hill. He's also primary research. He has a primary interest in understanding how chromatin regulatory mechanisms establish and maintain cellular identity, especially in the context of regenerative biology. The current research objective of his lab is to understand transcriptional and chromatin regulation of epithelial biology in the intestine and intrahepatic bioducts. Thank you for participating.
Thank you for that nice introduction. Can everyone hear me okay over there? Yeah. Yes? Okay, good. So again, thank you for inviting me and for that wonderful introduction. I have to apologize in advance for swooping in and out. I'm directing a GI initiative here, and we're having our first annual symposium, so it's a little bit of a crazy day over here in Atlanta. But it's really an honor to share some of our recent work with you, kind of an unexpected project that developed out of some of our work using intestinal organoids to model cycles of injury and regeneration. So as that was nicely said in the introduction, I kind of have this very long-standing interest in epithelial stem cell biology, and I did all of my training in the intestine. And, you know, I think we're all familiar with these concepts of stemness, but just to kind of orient everybody, you know, we define stem cells by their ability to self-renew, so divide and produce new daughter stem cells, as well as their ability to differentiate and produce all of the mature lineages that we see in a tissue like the intestinal epithelium. So kind of this homeostatic renewal of epithelial tissues is one thing that we're interested in. But then, of course, we know that our epithelial tissues are constantly exposed to insults like infection, injury, and inflammation. So they have to go through these cycles of injury, repair, regeneration, and return to homeostasis. And in this setting, we can have regeneration that's not driven exclusively by a pool of what we would call professional stem cells, but we can have different populations that exhibit facultative stemness and de-differentiate to maintain that epithelial tissue. And then, of course, we know, like over time, with chronic inflammation, chronic injury, it can increase the risk of tumorigenesis. And again, this can arise from either a professional stem cell population or these facultative stem cells that reacquire functional stemness through some regulatory mechanism. And so more and more, my lab has been moving towards understanding stemness as a property that's not exclusively restricted to a specific cell population, but something that can be accessed in a context-dependent manner by a bunch of different cell types. And so our primary focus is trying to understand the genomic regulation, so the interaction between transcription factors and the cis-regulatory landscape, and how these things can influence a cell to either enter, exit, or re-access a stem cell state. To do this, we use two model systems. The first is the intestinal epithelium. It's arranged as a monolayer of cells along a crypt and villus axis. We've known for many, many years that the intestinal stem cells reside at the base of these crypts and drive a process of proliferation and differentiation and migration up the villus axis. So this whole journey from crypt base to villus tip takes about 1 week in mammals. So our intestinal epithelium is constantly renewed. And because of that, we need a dedicated active intestinal stem cell pool. The other tissue in my lab has moved into, really when I transitioned to independence, are the intrahepatic bile ducts of the liver. The liver is really interesting because it's highly regenerative, but there's really no evidence for a dedicated stem cell pool. And we think that's because the homeostatic turnover in the liver is quite low. So this is a tissue where you have a robust regenerative response, but it's driven, we think, entirely by facultative stemness. So really the long-term goal for the lab is to try and understand, are there shared and distinct genetic programs that regulate stemness between multiple tissues with very different renewal characteristics. I'm actually going to focus solely on the small intestine today, so we'll talk about that moving forward. So I told you that the small intestinal epithelium has active intestinal stem cells. We know that these localize to the base of the crypts, intercalated between mature Panna cells, which is a secretory cell type that provides niche signals to the stem cell, and also secretes antimicrobial peptides to protect that crypt-based niche. We have very well described biomarkers now for active intestinal stem cells. The one that everybody thinks of that was the first is LGR5. We also know they express genes like OLFM4, ASCL2. There's a whole active intestinal stem cell gene signature now that's very well accepted. And so normally these cells will divide about once every 12 to 24 hours on average and differentiate either into secretory progenitors or absorptive progenitors, which then go on to produce the mature cells of the epithelium. We used to think that there was a +4 reserve intestinal stem cell. And when I started grad school, it was thought that this was a quiescent population that does nothing but wait around for injury to the active intestinal stem cell. And then it can, jump back into the cell cycle and maintain the epithelium. What we now know is that these +4 cells from the base of the crypt are actually early progenitors. So in the absence of injury, they will go on to differentiate into secretory lineages. And rather, when we get injury to that active intestinal stem cell pool, multiple different populations of early progenitors and even some mature cells like the Panna cell can de-differentiate and act as facultative intestinal stem cells. So of course, from a genetic standpoint, this is fascinating, right? How do you take a mature cell or a differentiating cell, and then rewire all of the gene regulatory programs that it's executing on the way to differentiation and pull it back to stemness. So that's one question we want to understand. The other question we want to understand is, after you get cycles of injury and regeneration, how does the epithelium return to homeostasis? That's actually poorly understood. And then, of course, are there long-term consequences to these cycles of injury and regeneration? So, things like chromatin-mediated transcriptional memory of prior injury. Is this something that over time can potentiate the epithelium to develop into tumors? So these are all questions that the lab is interested in pursuing. There's a number of different models that we can use to induce this facultative stemness. And the classical one is ionizing radiation, so gamma radiation. It's actually a pretty difficult protocol to do. It's pretty hard on mice. And it's just not very tractable for answering these questions about cyclical injury. So we turned to using doxorubicin. So doxorubicin is a chemotherapeutic drug that's been shown very nicely by Chris Decani's lab to induce injury to the intestinal stem cell. So what I'm showing you here is just a simple histology of doxorubicin and then progressive injury. When we look in vivo at LGR5 EGFP, so this is our active intestinal stem cell marker, we could see that by 24 hours, the active intestinal stem cells are largely lost. And that they take a few days to come back in vivo. Peter Dempsey's lab at University of Colorado has shown very nicely that in the setting of doxorubicin injury, you get lineage tracing from mature Panna cells. So this is kind of the gold standard evidence of facultative stem cell function. In a normal mouse without doxorubicin, there's lineage trace TD tomato signal restricted to the Panna cell. So these cells are not lineage tracing. After doxorubicin, they're able to give rise to clonal epithelial colonies, so showing that they are now functioning as stem cells. Some work has been done using intestinal organoids and doxorubicin injury, but it's all been restricted to very early time points. So Chris Decani's group, again, has shown that you can treat LGR5 GFP organoid with doxorubicin, and by live imaging, these cells will kind of eject those LGR5 GFP positive active intestinal stem cells into the lumen. So they're delaminating from the epithelium and dying. So it's some evidence that the organoids can reproduce active intestinal stem cell injury in response to doxorubicin, but no one's really looked at longer term injuries. So what we wanted to do is just very, very simply actually started this project for rotation students in the lab. And my idea is that we just want to, really just do a dose curve and optimize an organoid model of doxorubicin-based injury and recovery. We want to use this model in conjunction with our in vivo studies to understand epithelial autonomous mechanism of active intestinal stem cell to facultative intestinal stem cell cycling. And it's also, I thought, a platform it would be easier to, dose these organoids with doxorubicin, let them recover, and then do a secondary injury, and easier to do this in organoid models than in vivo. So the first thing we did is we set out with the goal of optimizing an organoid model of doxorubicin injury that would facilitate facultative intestinal stem cell mediated regeneration that was compatible with long-term survival, right? So we don't want to just kill the organoids by giving it a lot of drug. We want to kind of mimic what would happen in vivo, give a pulse of doxorubicin, withdraw it, and then have that organoid recover. So we plate organoids, we wait about 48 hours for them to establish in culture, and then we give them a 4-hour pulse of doxorubicin, after which we rinse that out and make sure there's no residual drug. We did a preliminary dose curve, and we found that 0.25 and 0.5 microgram per mil was the effective dose range that allowed organoids to survive. And this is what it looks like when we treat the organoids. Not surprising at all. By 48 hours post doxorubicin, you can see relative to untreated controls that are low and high dose doxorubicin induce a lot of apoptosis. Epithelial monolayer in the organoids gets a little thinner, a little less happy. And whereas our control organoids keep growing, proliferating, and forming nice budding crypt domains, the doxorubicin treated organoids are stunted in growth. We wanted to make sure that this was highly reproducible before we moved on. So Joseph Lee, who's a research specialist in my lab, did a really nice and pretty painful quantification counting many organoids from 4 mice, 3 wells per mouse. And we show that we have a reproducible loss of organoid survival that's also dose dependent. So this was important for us to have like a high and a low dose of doxorubicin. We can see 10 days post-dox that we still have some surviving organoids in all of our condition, and that higher doses of doxorubicin result in more organoid death, which is not really surprising. Next, we wanted to ask, are we actually inducing active intestinal stem cell injury and facultative stemness? So the easiest way to do this is by qPCR. We isolated RNA at 24 and 48 hours post-doxorubicin and calculated fold change in gene expression relative to our 24-hour untreated controls. So we could see that very clearly our canonical active intestinal stem cell marker LGR5, as well as OLFM4, which is another one that I mentioned. Both of these are significantly downregulated at both doses of doxorubicin. We don't really see a significant difference in the level to which these markers are downregulated, although it is trending further down with the higher dose of doxorubicin. Of course, we want to look at our facultative intestinal stem cell markers. So the two that are very well accepted in the field are Ly6A, which is also called SCA1, and CLU, which is a target of the YAP signaling that also has mechanistic function in vivo in facultative stemness. We could see for both of these that they're rapidly upregulated at 24 hours after doxorubicin. And that at 48 hours, they continue to be upregulated in the higher dose while they stay about flat with the lower dose of doxorubicin. So what we can take with this is that, as expected, and as we see in vivo, doxorubicin induces active intestinal stem cell injury, and at least by gene expression, triggers that facultative response. Now, because we're interested in how these organoids recover and how we can apply this model to studies of longer-term regeneration and cycles of injury and repair, we wanted to also look at further post-injury time points. So at 5 days, you can kind of see, hopefully by these images, this is a low magnification, but you can see some epithelial buds. So these are the crypt domains of our untreated control organoids, while all of the doxorubicin-treated organoids are very small and they kind of remain covered and obscured by the debris of apoptotic cells that we get from that initial injury. So accordingly, at 5 days, we see that both low and high-dose doxorubicin are significantly downregulated in terms of active intestinal stem cell markers and still express high levels of the facultative stem cell markers. And again, this is an effect that's more exaggerated in the higher dose of doxorubicin. When we look at 10 days, things are actually quite different. So we can see now pretty clearly by microscopy that the untreated control organoids have gotten very large and have many cryptobud domains. That's what we expect. This is also the point where control organoids start to get unhappy because they're overcrowding the well and really probably need to be passaged. What's interesting is from the doxorubicin injury, we can see these large cyst-like organoid. And so this is a very well appreciated morphology in intestinal organoids, where that cyst-like structure is associated with hyperproliferation, high levels of Wnt signaling, and high levels of active intestinal stem cell marker gene expression. And sure enough, that's what we see. If we look at qPCR at 10 days and compare our doxorubicin-treated organoids to our untreated controls, we could see that they're expressing significantly higher levels of the active intestinal stem cell markers. What's also interesting is that the facultative markers are kind of all over the place. So they don't reach significance because in each condition that we looked at, we had one well that was an outlier in terms of facultative stem cell marker gene expression. So what we think is happening here is we think that 10 days is this transition point where you've returned the organoid to an active intestinal stem cell state, and some wells, some organoids are still in the process of turning off those facultative injury response genes. So what we wanted to do now is we kind of have like this rough time frame laid out by gene expression for when we have transition between active and facultative stem cells. But we really wanted to nail down like the transition timing with a little more accuracy of when do we lose active stem cells, when do we gain facultative stem cells, and when do the active stem cells come back. And we thought, well, the best way to do that is to have a good biomarker and just live image and figure out these transition time points. So we turned to a pretty standard model in the field to do a pilot experiment. So we used LGR5 eGFP organoids, so they express a GFP reporter. What we did is it's kind of a rough experiment because we wanted to just see if we could pull this off before we did like a confocal. But you can see these like little stripes of very bright regions of GFP. So these are our LGR5 GFP high intestinal stem cells. Now, the tricky thing with this experiment is this allele is known to be mosaic. So not every LGR5 stem cell will have GFP. What helps us is that it tends to be clonally mosaic. So if you look at a mouse, some crypts are GFP positive, some crypts are GFP negative. So you isolate those crypts and you grow them into organoids, you can segregate your experiment based on GFP expression. So what we did is time point 0, we identified all of our GFP positive organoids, put them on an automated scope where we could set stage position and count the number of stem cells per organoid following the same organoids day to day. We treated these with doxorubicin and then counted as best we could the number of stem cells day over day. And the really surprising result from this pilot experiment is, as you would expect, control organoids that aren't treated are growing and their stem cell pools are expanding. But much to our surprise, we never saw a doxorubicin treated organoid that survived and lost all of its active intestinal stem cells. The only surviving organoids treated with doxorubicin always had at least one LGR5 high cell that survived day over day over day. This was really surprising to us. It kind of went against our expectation. But how do we really quantify that, right? I mean, we're following images as best we can by eye, but it's very difficult to make a quantitative argument with this allele because of the mosaicism and just because of the difficulty of trying to do this with imaging. We turned to another model that the lab uses, and this is a SOX9 GFP reporter that I helped develop in grad school and that we currently use in vivo to study chromatin dynamics. And basically what you need to know about this allele is that SOX9 is expressed broadly throughout the intestinal crypt. It's largely turned off in mature cells with the exception of some rare secretory lineages that are GFP high. And what we can do is isolate different levels of GFP expression to study different types of cells. So GFP negative are mature cells. The GFP sublow cells live at the neck of the crypt, and these are early progenitors. The low cells are the intestinal stem cells at the crypt base. And then these high cells are a mix of different secretory lineages and secretory progenitors. So we knew that we could grow organoids from the SOX9 GFP mice. We know that this reporter is non-mosaic. And we know that the organoids, just like in vivo, express different levels of GFP. So you can see in this little crypt domain here, we have our GFP negative pana cells, which are granulated on bright field microscopy, and our GFP positive crypt base. But no one had ever isolated these populations from organoids. So we don't really know if that reporter can work in vitro the same way it does in vivo. So we grew these organoids up, we fax isolated populations, and by qPCR, we can see that this model allows us to capture the same populations in vitro as it does in vivo. So what I will highlight here for the sake of time is that LGR5, or active intestinal stem cell marker, is significantly upregulated in the SOX9 GFP low. That's the population we're interested in. And then mature markers like fatty acid binding protein 1 are downregulated, as well as chromogranin B downregulated in the lows. So moving forward, the important take home is that SOX9 low is the active intestinal stem cell population in this model. So now what we can do is we can treat these organoids with doxorubicin. We can get around the mosaicism issue with LGR5 GFP, and we can apply flow cytometry to quantify stem cell dynamics. So first we wanted to look by eye. So we treated with a dose curve of different doses of doxorubicin. We also treated with the very high dose doxorubicin that's incompatible with organoid survival. And like you would expect, we see that our 0.5 microgram per mil doxorubicin reduces the number of GFP positive cells. We expect that because doxorubicin is going to target the cycling cells, it's going to target the stem cells, and that's what's marked by GFP. What's kind of cool is if we go to the higher doses, and especially this highest dose that we tested, where you see this organoid does not survive very long after doxorubicin, it's very difficult by eye to find any SOX9 low or sublow cells. All we see are these negative cells and the high cells, which are mature cell types, non-cycling and probably resistant to doxorubicin. So of course we want to quantify this by flow, like I said. So we see that by flow analysis, our 0.25 and 0.5 microgram mil per dose of dox decrease significantly the number of SOX9 low intestinal stem cells, but they don't totally eliminate those populations. So this is consistent with what we see by microscopy for LGR5 GFP. One thing that's interesting here, just like the LGR5, is that in our control organoids from 24 to 48 hours, the proportion of active intestinal stem cells increase like we would expect from a growing organoid, but they do not change in doxorubicin treatment. So this suggests that doxorubicin is not eliminating the active intestinal stem cell pool, but stunting its expansion. I'm not showing the data here, but the very, very high dose of doxorubicin shown here by flow cytometry nearly completely eliminate the SOX9 low populations, and we don't see very many remaining. Finally, a postdoc in my lab, Antoine Glizes, wanted to functionally validate that we're having a loss of stemness associated with doxorubicin treatment. So he took organoids, treated them with, again, our low, what we're calling low and high dose doxorubicin, and then the very high dose that's incompatible with organoid survival. He quantified the decrease in survival at 24 hours post-doxorubicin, and then he passaged these organoids. So our thought was if we dissociate these organoids to single cells and passage them into high wind conditions that favor organoid formation from clonal single intestinal stem cells, we predicted that our lower dose of doxorubicin would retain some functional capacity, some functional active stemness that would allow those cells to produce organoids, where the higher dose would lose that. And that's pretty much what we see. So our organoids that are not treated at 5 days have produced these nice typical intestinal organoids from single cells, where higher doxorubicin treated like rapidly lose this, but it's still retained to some degree with our lower doses of doxorubicin. We also noticed that all of the doxorubicin-derived organoids on passaging have this kind of defective morphology. So it's some functional evidence on top of all our biomarker work that we have these residual active intestinal stem cells, and that the ability to be passaged is nearly totally lost at doses of doxorubicin that are inconsistent with organoid survival. So how do we really functionally test whether these residual stem cells are contributing to organoid survival after injury? For that, we turn to a LGR5 2A DTR allele that was kindly shared by Nick Barker's lab in Singapore. Basically, this allele expresses a diphtheria toxin receptor, specifically in the LGR5 positive intestinal stem cells. Mice, unlike us, are highly resistant to diphtheria toxin. So you can give a very, very low dose, and if a single DT molecule is capable of entering that LGR5 cell that's expressing the receptor, then it will kill that cell. What's nice about this model is it allows us to ablate cells without triggering the type of inflammatory response and DNA damage that you would get with something like irradiation or with doxorubicin injury. So just looking in vivo, these are LGR5 positive cells by in situ. This is a control mouse. This is a mouse that expresses that diphtheria toxin receptor, and you can see the DT treatment eliminates that LGR5 active intestinal stem cell population. So again, we wanted to pulse organoids with DT, show that we could get a significant reduction of active intestinal stem cell markers. This is indeed what we see with a 1, 2, or 3 hour pulse of DT. We did then also want to validate that the diphtheria toxin is not causing organoid death in and of itself. And so what we see when we pulse organoids with DT is we do get apoptosis and we get kind of disruption of normal organoid morphology, but we never fully lose organoids even at 5 days when they start really exhibiting signs of recovery. So now we have a model where we can ablate active intestinal stem cells without inducing DNA damage and not affect organoid survival. So the obvious experiment to do here now is to repeat our doxorubicin injury, wait 24 hours where we know we have those residual active intestinal stem cells, and then pulse them with diphtheria toxin to ablate the active ISCs. When we do this experiment, we see that intestinal stem cell ablation accelerates organoid death and results in a total loss of survival, even at our lower dose of doxorubicin, which, normally without the diphtheria toxin survives at about like 30 to 50%. So here you can see our untreated controls, organoids treated with diphtheria toxin only in blue, organoids treated with doxorubicin only in red, and organoids treated with doxorubicin and then stem cell ablated in purple. And we do not see any organoids that are capable of surviving if we ablate the stem cells after doxorubicin. So what does this all mean, and why is this relevant for drug modeling and organoid biology? So we know in vivo, or at least in homeostasis, that we have active intestinal stem cells that are driving turnover of epithelial cells and homeostasis. And we know that this is accurately modeled in organoid cultures. It's pretty well accepted in the field now. When we have injury, we know in vivo that we get loss of active intestinal stem cells and multiple facultative stem cell populations that are able to regenerate the epithelial lining. So we know that in organoids, we can model the AISC loss and the activation of facultative stem cell genes. But what we've shown here is that we can't reproduce the facultative stem cell function, right? Those genes are coming on, but functionally the cells are failing to maintain the epithelium if you don't maintain the active intestinal stem cell pool. So our questions here moving forward and why we think this is important is a lot of people are using organoids and even primary patient organoids to do drug screens and try and identify the best therapies for things like colorectal cancer. Because cancer is a stem cell driven process, we think it's very important to understand the potentially different dynamics between stem cells in vivo and an organoid system. And there's a lot of evidence that I don't have time to talk about supporting a role for non-epithelial cells that aren't present in our organoids in supporting facultative stemness and regeneration. And so we think that this platform now where we can injure organoids with doxorubicin and ablate the stem cells with DT is a platform that'll help us maybe recapitulate some of these non-epithelial signaling sources and build better organoid models for facultative stemness. So with that, I will thank you all for your time and attention and the invitation to share our work. And I want to acknowledge both Joseph Lee, a research specialist, and Antoine Glizas, a postdoc in the lab, who are first authors on a manuscript we just posted on bioRxiv. I think it actually went up today. And I'll be happy to answer any questions if you have them.
Thank you very much, Evan, for your nice presentation. We are now opening for question and answers. I would like us to invite Natalia to come here, Lida. And I open to the audience if someone would like to ask some questions. And Tatiana, you can also check, please, at YouTube.
I think I may have to sign off because I have to run to our symposium in a couple of minutes.
Okay.
So yeah, sorry to dip out. Thank you for having me, and enjoy the rest of the
Thank you very
symposium.
much for your kind presentation. Thank you, Adam.
Thank you. Bye-bye.
Acho que agora a gente pode falar em português. Fica mais fácil. Eu vi que a Natália ficou no intervalo conversando com algumas pessoas e a gente queria aproveitar que vocês aproveitassem essa oportunidade, já que nós temos, né, a Natália, que trabalha com isso, que ela pode ajudar, de repente, vocês a desenhar os projetos. Nós colocamos ela no nosso grupo justamente para isso, para facilitar na hora de desenhar e escrever os projetos, fazer os orçamentos, e porque ela já tem experiência, é um projeto caro, então não dá para a gente errar muito, né? A outra coisa, a gente convidou todos esses pessoal da Emory, porque a Leda já vem trabalhando com o pessoal da Greiner, e ela também conseguiu adquirir um pouco de experiência trabalhando com essa metodologia, e seria uma forma de compartilhar os erros, né? E os troubleshootings, né? O que eles fizeram para sanar esses erros, né? Porque, novamente, como eu disse, como é caro, a gente tem que primeiro ver o que os outros fizeram, ver onde eles erraram, para tentar evitar esses erros e diminuir, né, os nossos prejuízos. Alguém tem alguma pergunta? Se não, vou começar uma para a Natália. Eu vi que muitos apresentaram sobre cocultura, tanto de células, como a Leda apresentou cocultura de células e de vírus, né? Existe algum limite para fazer cocultura de células, limite de células diferentes? Existe um número mágico? A cocultura ajuda? A gente sentiu que ajuda, né? Você poderia comentar sobre isso?
Obrigada pela pergunta. É uma pergunta muito relevante e interessante de ser feita, porque, a princípio, eu não vejo um limite, porque a ideia da cultura 3D é você desenvolver um modelo que represente o máximo possível que você tem em vivo. Eu sei de exemplos que eles fizeram mais de 4 tipos celulares, então foi possível montar. Era um bronquíolo, o modelo, é um artigo que está publicado. Ele não é tão recente, já tem 10 anos, mas eles foram bem-sucedidos na realização desse modelo. Eles conseguiram estabelecer um modelo bem funcional para estudar, inclusive, modelos de infecção e eu acho que tudo é padronizável. Claro que quanto mais células você coloca, mais complexidade você traz também para validar e estabelecer o seu modelo. Mas eu, a princípio, não vejo uma limitação do que pode ser feito, né? Muitos trabalhos fazem a própria diferenciação das células e aí você obtém a organização dela naturalmente também.
Eu lembro que ano passado, quando nós, não, no retrasado, quando nós fizemos nossa parte hands-on, que a gente recorrer atrás dos fibroblastos, né? Que, na verdade, até fazer cocultura, ela até pode ajudar, né?
Sim, exato. O fibroblasto, em algumas situações, né, a gente tem células que produzem menos matriz, então a associação com outros tipos celulares ela pode colaborar. Além de você trazer mais representatividade, né, de você ter outros tipos celulares ali presentes em enriquecer a sua amostra em sinalização, em moléculas presentes ali, você também tem essa contribuição para o estabelecimento da matriz extracelular. Então, quando a gente tem algumas circunstâncias como essa, uma das abordagens que nós fazemos é, quando possível, para o modelo que está sendo utilizado, também incorporar um outro tipo celular, como, por exemplo, o fibroblasto. Ele vai reforçar ali o estabelecimento da sua cultura. Aí entra a parte de padronização, que eu acho que a Leda já tem muita mais experiência ainda, na parte de padronizar e de acertar a quantidade, proporções que você vai utilizar de cada tipo celular. Porque isso vai variar também de modelo para modelo, por exemplo.
Tem uma pergunta, se não tem mais... Ah, lá.
Olá, até assim, acho que na linha, né, no mesmo raciocínio do que a Natália está dizendo, pensando nesses desafios, acho que vai mais além ainda quando a gente trabalha com células de paciente, né, amostras de paciente. Então, não sei se vocês teriam até alguma dica ou até alguma experiência prévia para compartilhar nesse contexto de organoides a partir de amostras de pacientes, por exemplo, tumores, enfim, qualquer outro exemplo.
Alô, alô? Pode? Ah, eu mostrei, né, mas eu não mostrei mostrei de camundongo, né, na verdade. E se você conseguir o tecido, você tem que tratar ele um pouco, fazer uma preparação, como eu mostrei, dissociação do tecido e também fazer um tratamento enzimático para poder preparar ele para fazer o organoide. Mas não é difícil, como eu falei, ele parece, o que eu mostrei, parece um liquidificador. Eu acho que aqui deve ter um processo que você prepara o tecido para fazer o organoide. Aham, aham. Sei. Com os organoides. Aham.
Desculpa. E você acha que, então, a partir tendo as amostras digeridas, acho que o processo de formação do organoide não vai ser um problema. Você acha
Não,
que não é
vai
um
ser um
problema?
problema, não. Desde que você prepare bem digerido, como você falou, você consegue fazer o organoide rapidinho.
Tá.
Principalmente com o sistema deles é mais fácil. O matrigel eu acho um pouco mais difícil. Eu vi uma pessoa no laboratório que trabalhou bastante com matrigel, tem uma outra pessoa que trabalha que demora assim umas 3, 4 semanas para lidar com matrigel. Esse é um pouco mais fácil, mas de novo você tem que fazer uma digestão, você tem que preparar a amostra.
E
Se
depois,
você já tem o sistema, aí é fácil de fazer o organoide.
depois do co-cultivo também, você acha que
O
que
co-cultivo
é
é super
tranquilo?
fácil. Como a Natália falou, você tem que saber um pouquinho da proporção, mas não é difícil, não. Não é difícil, não. E como você coloca, primeiro você faz o organoide, depois você põe o co-cultivo em seguida e ele, ele abraça o organoide, sabe? E como ela falou, os nutrientes que, por exemplo, o fibroblasto traz, é bom para, nesse caso, para o tecido de fígado, por exemplo. Então funciona super bem. Eu mostrei algumas fotos né, em volta. Por exemplo, eu usei o CD4 e foi bom, porque o CD4 não é célula em suspensão, né? Também eles não ficam muito felizes, mas eles ficaram no formato de. E é muito importante também o tipo de placa que você usa para não aderir a, o organoide, o esferoide, porque você não quer que adere à superfície, né? Você quer que fique em suspensão, né? Isso é importante também. Mas ela pode dar as dicas super.
Tá bom.
E
Obrigada.
se precisar qualquer coisa também, a gente dá a dica, dá protocolo, como a gente fez, tudo.
Legal, legal. Obrigada.
Também. Acho que indo na linha do que a professora Leda já falou perfeitamente, você trabalhar direto com a célula do paciente para o organoide, você tem uma vantagem, que essa célula não vai sofrer tanta modificação se você já incorpora ela num sistema tridimensional, por exemplo. Ela já vai estar num ambiente mais enriquecido do que você passar, de repente, correr o risco de perder ou descaracterizar a sua célula, cultivando ela por muito tempo em 2D. por exemplo. Então, eu acho que é uma das vantagens que você tem também já de trabalhar direto com o salo do paciente. Provavelmente você vai precisar, às vezes você tem uma quantidade de amostra pequena, né? E aí você precisa guardar e expandir ela um pouco, mas você tem as vantagens de não perder tanto da morfologia nem da característica que ela tem já vindo do paciente, por exemplo.
Tem uma pergunta aqui. Giovana Caetano, perguntando se é viável utilizar a cultura de célula 3D para edição genética utilizando a metodologia de CRISPR. CRISPR.
Sim, você pode usar o 3D, o esferoide, e fazer o CRISPR. O CRISPR não interfere em nada. Aliás, ajuda até. Ajuda,
E só voltando à questão de usar material biológico direto de pacientes, o Flávio montou o grupo com o André Barateiro justamente para coletar, para poder trabalhar com a amostra de pacientes. Eu acho que, na verdade, o mais difícil não é a metodologia em si, porque se você, por exemplo, se você usou de um animal, treinou no animal, né? Depois você consegue reproduzir com o ser humano. Eu acho que o mais difícil é a parte burocrática, passar no comitê de ética, ver o termo de consentimento, pegar a assinatura do paciente, agendar horário com o cirurgião, com o patologista. Então, acho que a parte logística acho que é mais trabalhosa do que a parte laboratorial. Tá?
E, assim, nós estamos fazendo esse projeto, na verdade, é um projeto onde nós vamos aproveitar o resto que não é utilizado na análise do tumor, né, quando se faz a ressecção, e para fazer a cultura. Mas realmente, como falou a professora Suzane, nós estamos já 2 meses fazendo o termo de consentimento livre esclarecido. Graças a Deus acabamos agora. Já tem toda essa parte, mas eu acho que temos que ir. É o que estamos fazendo, né? Pensando nessa logística para obter esse material. Afinal de contas, esse material vai ser descartado, então é um material precioso para que a gente possa fazer essas pesquisas, né? Alguém tem mais alguma pergunta? Algum questionamento mais?
Eu tenho mais uma pergunta. Já que não tem pergunta, vou aproveitar e tirar as minhas dúvidas. Eu vi que uma das apresentações eles mostraram que formou um donuts, né? Isso por causa do formato do ímã. Então, é possível a gente desenhar o formato que a gente quiser
No ímã.
no ímã?
Talvez o Glauco saiba dizer isso melhor do que eu, porque foi ele quem desenvolveu a metodologia. Eu não sei se é possível fazer outros formatos. Eu sei que eles conseguiram fazer no formato de anel. Não foi algo simples, talvez, de ser feito, porque a gente está falando de campo magnético, então tudo isso tem que ser levado em consideração, provavelmente, né, na hora de desenvolver o ímã. Quando a gente começou, as metodologias foi feita a levitação primeiro, então eu sei que depois eles conseguiram otimizar para fazer o bioprinting, no caso da magnética, e o anel também. Mas eu não sei se é possível fazer qualquer formato. Então, realmente, a gente pode levar essa pergunta amanhã novamente para o Glauco.
Então, tá joia. Obrigada.
Fazer essa ideia, se não tiver ainda, né? Eu acho que é interessante.
Não é? É uma ótima oportunidade, por que não?
Eu acho que é um dos motivos aqui da nossa Vamos desenvolver se não tiver. Muito bom, muito bom. Então, essa sessão, então, estamos encerrando agora. Vamos ter aí um período de almoço. Aqui embaixo também tem almoço na nossa cafeteria, aqui no térreo, tá? Quiserem almoçar, podem ir lá. E voltamos
8h30.
amanhã, às 8h30, estaremos aqui de novo. Muito obrigado pela presença de todos. Esperamos vocês amanhã. Uma coisa só, quando vocês forem fazer perguntas, se apresentem falando o nome. Gostaríamos de saber até seu nome. Ah, muito legal, muito legal. Sejam bem-vindos, então, amanhã de novo e vamos em frente no nosso curso aí. Muito obrigado.
Obrigada.